Welcome to the Deep Dive. Our mission, as always, is pretty simple. We take a huge body of work, and today we're talking about the absolute bedrock of medical science, and we just distill it down to the essential, high-yield info you need to, get it right away.
Right, and today we're really leaning on a giant in the field, Ganong's review of medical physiology.
The 26th edition, specifically.
Exactly, and you have to give a nod to Dr. William Ganong himself. His work, it fundamentally shaped how generations of doctors think about the human body.
So our goal is the same as his. Make it clear, make it scientific, and make it relevant. We're going from the tiniest cell membranes all the way up to huge hormonal systems.
Yeah, and we'll see what happens when those systems go wrong.
Absolutely. By the end of this, you should feel pretty fluent in these core concepts.
Huh.
Okay, so let's start at the very beginning. The body's basic organization, where's all the water?
Yeah.
How is it, compartmentalized?
Well, what's so fascinating is how much of it is actually inside our cells.
Oh, really?
Yeah, 2/3 of all your body water is the intracellular fluid, the ICF. That's about 40% of your total body weight.
Wow. Okay, so what's left is on the outside.
Right, the extracellular fluid, or ECF. That's the other third. And that's split between the fluid bathing the cells, the interstitial fluid, and, what's actually in your blood vessels, the plasma.
Which is actually a pretty small percentage when you think about it.
Only about 5% of your body weight.
So with water moving between these spots, we need a way to talk about the forces driving it. Let's jump right to the clinical term, osmolality.
Right, osmolality. It's just the concentration of particles in a solution, and that's what pulls water across a membrane.
But in the clinic, you hear tonicity a lot more.
We do, because tonicity is more practical. It's the osmolality of a solution relative to your blood plasma. It tells you what effect that solution will have on your cells.
And this is where it gets really important with IV fluids.
Mm-hmm.
So explain that classic example.
Mm-hmm.
How can a fluid start out as isosmotic, but then act like a hypotonic fluid?
Ah, yes, the 5% glucose solution, or D5W.
Yeah.
This is such a key concept.
Yeah.
So when you first hang the bag, it has roughly the same particle concentration as plasma. It's isosmotic. But, and this is the key, your body immediately starts metabolizing that glucose.
It just uses it for energy.
Exactly. And once the glucose is gone, what are you left with?
That's water.
Just free water. So the net effect is that you've infused a hypotonic solution. That water is going to move into the cells and make them swell. Totally different from 0.9% saline, where the salt stays outside the cells.
That's a great clinical pearl.
Yeah.
So that metabolism, let's talk about the engine that drives it all. We know the cell membrane is there, but what's the core energy pathway?
It all comes down to the citric acid cycle. You might know it as the Krebs cycle or TCA cycle.
Right.
This is the final common pathway where we oxidize everything, carbs, fats, proteins, down to CO2 and water. But it has one absolute non-negotiable requirement.
Oxygen.
It needs oxygen. Without O2, that cycle shuts down completely. It's the fundamental link between breathing and making energy.
Okay, so from making energy to using it, let's talk about how our neurons and muscles fire.
Yeah.
How do they create that fundamental voltage, the resting membrane potential?
Well, the RMP, which is usually around, say, minus 70 millivolts, is mostly set up by potassium.
K plus B.
Yep. You have a ton of potassium inside the cells, so it naturally wants to leak out through these special channels.
Following its concentration gradient.
Exactly. But as that positive potassium leaves, the inside of the cell becomes negative, and that negative charge starts pulling the positive potassium right back in.
So you have two opposing forces.
Precisely. And the resting potential is the point where those two forces, the chemical push out and the electrical pull in, are perfectly balanced.
But something has to maintain that balance over time, right? It can't just be passive.
You're absolutely right. That's the job of the NA-KAT-PACE pump, the sodium-potassium pump.
And we call it an electrogenic pump. Why is that?
It's electrogenic because it moves an unequal amount of charge. For every cycle, it pumps 3 positive sodium ions out. For every 2 positive potassium ions it pumps in.
So there's a net movement of 1 positive charge out.
Exactly. That contributes a little bit to the negative charge inside, but more importantly, it maintains those concentration gradients that the whole system depends on.
That pump is just heroic. Okay, so that's the foundation. Now, how do cells talk to each other? Let's focus on the big one, the receptor class that dominates pharmacology. G protein-coupled receptors.
GPCRs, they're probably the single most important drug target family we have.
How many drugs target them?
Something like 40% of all prescription drugs, they're involved in Vision, smell, mood, blood pressure, just about everything.
So how do they work in a nutshell?
They're like a middleman. A signal like a hormone binds to the outside, the receptor changes shape, and it activates a G protein on the inside. That G protein then goes off and activates something else, usually an enzyme.
Which then makes a second messenger.
You got it. It's an amplification system.
So give us a really high-yield example of one of those messenger systems.
Oh, a classic one is the pathway that generates IP3 and diacylglycerol, or DIR8.
IP3 and DAGI.
Right. The IP3 floats over to the endoplasmic reticulum and basically opens the floodgates for calcium release from inside the cell.
And that burst of calcium does what?
Everything. It triggers muscle contraction, neurotransmitter release, you name it. It's a huge, powerful signal.
Okay, speaking of neurotransmitter release, let's use the neuromuscular junction as our real-world example. Nerve meeting muscle.
Perfect example.
And we can look at two diseases here, these channelopathies, that show a really neat contrast between a presynaptic and a postsynaptic problem.
Yes, this is so important for diagnosis. Let's start with myasthenia gravis.
Okay.
In myasthenia, the problem is postsynaptic. The immune system makes antibodies that attack the acetylcholine receptors on the muscle itself.
So the target is damaged.
Exactly. The nerve releases plenty of acetylcholine, but there aren't enough healthy receptors to create a strong signal. That's why you get that characteristic muscle fatigue.
Now contrast that with Lambert-Eaton syndrome, LEMS.
LEMS is the mirror image. It's presynaptic. The autoimmune attack is against the voltage-gated calcium channels on the nerve ending.
Ah, so it stops the signal from even being sent properly.
Precisely. Calcium has to rush in for the nerve to release acetylcholine. If you block those channels, you get a weak signal because not enough neurotransmitters released in the first place. The muscle is fine, but it's starved for a signal.
Okay, we've gone from the single synapse. Let's zoom out to the whole system, the endocrine system. It's all about these intricate negative feedback loops, right?
Absolutely. The classic model is hypothalamus to pituitary, and then the pituitary controls the target gland.
And a perfect example is the thyroid axis.
The thyroid axis is beautiful. You have thyroid hormones, T4 and T3, which set our overall metabolic rate.
Our body's thermostat?
Pretty much. And the pituitary releases TSH, thyroid stimulating hormone, to tell the thyroid gland to work. But the genius is in the feedback.
The negative feedback.
Exactly. As T3 and T4 levels rise in the blood, they go back and tell the pituitary and the hypothalamus to quiet down, to stop releasing TSH.
It's a self-regulating system.
Perfectly self-regulating. It keeps your metabolism right in that sweet spot.
Now, here's a great high-yield fact about thyroid hormone that isn't just about metabolism. What does it do to lipids?
This is a great one. Thyroid hormones lower your circulating cholesterol levels.
How do they do that?
It's actually a separate effect from the calorogenic one. They work by increasing the number of LDL receptors on your liver. More receptors means the liver can pull more bad cholesterol out of the blood.
So important when you're thinking about things like hypothyroidism and high cholesterol.
Very important connection.
Okay, let's switch from metabolism to fluid volume. The big one here is the renin-angiotensin system.
The RAS. Yeah, this is your body's #1 defense against low blood volume or hypovolemia.
It all culminates in one powerful molecule, right?
Yeah, it does. Angiotensin II. This molecule is a master regulator. It defends your blood pressure and fluid balance with really a four-pronged attack.
Okay, what are those four pillars?
First, it's a potent vasoconstrictor. It squeezes blood vessels to raise blood pressure immediately.
Right.
Second, it tells the adrenal glands to release aldosterone, which makes the kidneys hold on to salt and water.
Okay, so constriction and retention.
Third, it tells the pituitary to release vasopressin, or ADH, which makes the kidneys hold on to just water. And fourth, and this is crucial, it acts on your brain to make you thirsty.
Wow, so it constricts, it makes you retain fluid, and it makes you go get more fluid.
A completely integrated, powerful response to keep you from going into shock. It's remarkable.
So finally, let's talk about what happens when this kind of metabolic control just completely fails. I'm talking about severe insulin deficiency, diabetic ketoacidosis, or DKA.
Yes, DKA is a physiological cascade. It's a true medical emergency, and understanding the steps is key.
So walk us through it. Where does the disaster begin?
It starts with a two-part glucose problem. Without insulin, your cells can't take up glucose.
Okay.
Then your liver thinks you're starving, so it starts making even more glucose. The result is just profound hyperglycemia, sky-high blood sugar.
And then the kiddies get overwhelmed.
They do. When your blood sugar gets too high, you start spilling glucose into your urine. That's glycosuria. and glucose pulls water with it.
An osmotic diuresis.
A massive osmotic diuresis. You're losing tons of water and electrolytes, which leads to severe dehydration and that intense thirst we call polydipsia.
But inside, the cells are still starving for glucose.
Exactly. So your body panics and turns to its emergency fuel, fat. You start breaking down fat like crazy, producing huge amounts of free fatty acids.
And the liver turns those into ketones.
Right, into ketone bodies like acetoacetate. And the problem is these ketones are acids, organic acids.
So that's the acidosis part.
That's the ketoacidosis. Your blood becomes acidic. And that combination, the severe acidosis, the dehydration, the hypovolemia, it depresses your brain function. It can lead to coma and death. It's a terrifying cascade. Which brings us to a really critical clinical question. When you treat DK with insulin, why do you have to be so careful about potassium?
This brings us full circle right back to that NAT-KATPase pump.
The electrogenic pump.
Yep. Insulin is a major stimulator of that pump. So when you give a patient a big dose of insulin, you're revving up that pump in every cell in their body.
And that pump pushes potassium into the cell.
It shoves potassium from the blood into the cells. So even if the patient was already low on total body potassium from the diuresis, their plasma level might look okay at first. But the moment you give insulin,
It's all going to get sucked out of the blood.
it plummets. You can induce a sudden, fatal hypokalemia, low potassium in the blood, which can cause lethal cardiac arrhythmias. It's a classic example of how the treatment itself can create a new danger if you don't understand the underlying physiology.
Wow. That was a really powerful journey. We went from, a single pump in a cell membrane all the way to a full-blown metabolic catastrophe. We really covered a lot of ground, from tonicity and fluid shifts to GPCR signaling and then these huge interconnected feedback loops.
And it really shows how it's all linked. You can't understand DKA without understanding the NK pump.
That's it, exactly. It's not about memorizing facts, it's about seeing the connections. That's what understanding physiology is all about.
All right, as we promised, we have a final thought for you to chew on to really lock in this knowledge.
Right.
Let's circle all the way back to fluids. So the review question is this. We said that angiotensin II is released when you have low blood volume, right, to defend your extracellular fluid, your ECF. Now, considering that angiotensin II also triggers the release of vasopressin, or ADH, which makes your kidneys reabsorb pure water, not salt, what effect does this water retention have on the osmolality of the ECF, and what does that, in turn, do to the volume of the intracellular fluid, the ICF?
It's a great question.
Think about it. What happens when you add pure water to the ECF? Where does that water ultimately want to go? Connecting that huge hormonal cascade back to basic osmosis will really solidify this for you. That wraps up this deep dive. We hope this knowledge serves as a powerful foundation for everything you study next.